So now we'll come to the hands-on part, the sample prep, uh, where I will have Keith who will talk to you shortly about uh, what to do for this sample preparation part. Uh, and especially if you remember from the couple of sessions we had, in solution digestion or in digestion, what you have been doing, that pretty much remains same here as well. Because ideally, what you want the peptides to be prepared. But on top of the what peptides you're preparing, it's good idea for you to think about how you are going to do the assay and what you should add in the sample which could help you to track your assay. And these are the points which Shalini and Meher are going to talk to you now in terms of the sample preparation part.
Now, as Sir has already given an introduction and brief description about targeted proteomics and its workflow. So now we will be showing me and Shalini will be showing you about the sample preparation, like how to reach up to these samples that can be further analyzed as targeted proteomics. So, as in the previous live session, we have already shown the basic workflow of preparing peptides. So, this is the similar kind of workflow that we are using for sample preparation in case of this targeted proteomics. So, here we can see that these are the 4 or 5 basic steps prior to the mass spectrometry. So, here, as we can say, the first step is the reduction. So Shalini will show you how to do reduction. So these we have taken these samples. Initially, these are the protein samples. So firstly, what will be done? This reduction. Firstly, this reduction will be done. So Shalini is adding this reducing agent, that is the TCEP, this carboxyethyl phosphine, to the protein sample for reduction. So what this TCEP does, it reduces the disulfide bonds. It breaks the disulfide bonds of the between the polypeptides, so that those disulfide bonds can be broken, and these polypeptides, they will be easily accessible for the protease to act. So for this TCEP to act, we have to keep it at 37 degrees for about 1 hour. So now this TCEP is added, and after the 1 hour incubation, that reduction will be done. And now we are taking those reduced samples, and next what the step is alkylation. So this alkylation, it is done by using IAA, that is iodoacetamide, and this suitable volume of this iodoacetamide is added to the reduced sample. So what this alkylation does, it basically blocks the thiol groups of cysteines, so that these reduced disulfide bond, they did not reform. So we don't want them to be reformed again. So for that we are blocking those disulfide bonds from reforming, and for that we add this iodoacetamide as alkylation. So as this reaction, it is a light sensitive reaction, and we have to keep it for its proper action. We have to keep it in dark for around 10 minutes. This dark incubation will be done after this addition of this IAA. And then when this IAA is this alkylation is done, after that the next step, which is the main digesting step, is that the trypsin digestion comes. And in here we are using this trypsin enzyme for the digestion, but there are other enzymes also which are used for the digestion process. And for this trypsin, we require around 17 hours incubation at 37 degrees. So after addition of trypsin, we keep the samples at 37 degrees for around 16 to 17 minutes. So after this suitable incubation, we will get the protein digest, that are our desired size peptides. So when we get the desired size peptide, the peptides are dried, and then when we require them, they are again reconstituted. So, but the main important step here comes is that when we get these desired peptides, they are as by proceeding with a similar workflow, we obtain these peptides, and after this, these peptides need to be further cleaned up. Desalting is done. So when these peptides are cleaned up, so prior to mass spectrometry, before subjecting them to mass spectrometry, one more step is done. As here you can see that it's like spiking of heavy label peptide to our sample. So what is done? These clean peptides, they are spiked in with the synthetic peptides which are heavily labeled. So these synthetic peptides, they are exactly similar in their physical properties with our the target peptides. So the only difference is that these are labeled at a particular amino acid. So here, in our case, but synthetic peptides we are using, they are labeled at RNA. So these label peptide, what they do, they basically help in the quantification, so that we can quantify our target peptide in a sample. So when this labeling, this spiking is done, so after that we subject these samples for mass spectrometry for the targeted proteomics.
Hello. Uh, so this was the sample preparation part, and I hope by now you are all excited to take a look at the different approaches that Sir has already spoken about, that is SRM and PRM. So I'll be taking you through the technique of selected reaction monitoring, also called multiple reaction monitoring. So let me just share these slides with you. So once the sample has been prepared, we move to the main part of the lecture, that is the SRM and PRM. So I'm sure Sir has already spoken about the the need for targeted approaches and why exactly were global proteomic approaches not sufficient to answer many questions in the proteomics field. So here depicts the reason why targeted approaches came into picture. So as you can see here, this is a very good example to help you understand the need for targeted approach. This is the shotgun approach or the global proteomics approach that you have learned about in the previous lectures. So here you see a shooter aiming at a flock of birds, and on a lucky day he would hit the target, and that is pretty much the target, right? So in case of a shotgun approach, you basically are looking in the dark for proteins uh, uh, proteins which are involved in answering the problem that you're looking at. However, this may not be the actual picture every time. So, based on available literature, you can come up with different proteins that may be of relevance in the study. Right? So, the other side of the proteomic approach, that is the targeted approach, as opposed to the shotgun approach, is a much more selective approach wherein you know the targets beforehand and you are detecting them in a bit to quantify the abundance of this protein in the sample of interest. So the major application of targeted approaches have been in understanding the post-translational modifications and finding the levels of different proteins in the diseased patients. So this slide gives you an overview of the difference between the major difference between the shotgun and the targeted approach. Uh, I would also like to add that both the approaches are complementary and that none of the methods alone can help solve the problems that we face in the field of programming. The potential applications, as I had already spoken about, there are many different applications of this, and depending on the need of the experiment, the particular workflow can be followed in the targeted approach. Moving on to the instrumentation part of the selected reaction monitoring, the workhorse for SRM approaches, as Sir mentioned, has already mentioned, is the triple quadrupole instrument. So, looking at the diagram, you get a fair idea that here there are two quadrupoles, that is Q1 and Q3, which are in tandem, and they are separated by a collision cell that is labeled Q2. So, when I say triple quadrupole, uh, it doesn't mean that there are three quadrupoles. It is just that there is a Q1 separated by a collision cell followed by another Q3, right? So the basic nature of the mass analyzer, that is quadrupole, is that it acts as a mass filter. Okay, so in an SRM approach, one needs to have an idea as to the mass to charge ratio of the parent ion followed by the daughter ions which are formed, right? So this slide tells you about the principle involved in an SRM approach. As you can see, we have the Q1 mass filter and the Q3 mass filter separated by a collision cell, and both these act as a mass filter. The most important terminology that is used in SRM experiments, and you would come across this term whenever you read papers, is the term transition, as you can see here. And this is actually the Q1 to Q3 pair that is specific to a fragment under study, right? I would get to this in the next slide. It would become very clear there as to what exactly is a transition. So let us talk about this particular peptide sequence. As you can see, this is a big peptide with 12 amino acids in it, and our sequence under study is the one in the to the left of the table. As you can see, this is the same sequence which I have, which appears 4 times here. And if you can just visualize the instrument as I speak, the quadrupole 1 is set to a value of 678.83. Okay, so this is the Q1 mass filter which I have set for my experiment. However, when this peptide under study fragments, it can give rise to different fragments based on where the peptide bond has got cleaved. Like, say, example, if this particular peptide bond between between P and T gets cleaved, then it would result in 2 different fragments, right? And each of these fragments has a different mass to charge ratio. So basically, in an SRM experiment, what you are instructing the instrument is to look at a Q1 and a Q3 pair, right? So this is a transition. In this particular example, there are 4 transitions being studied for one peptide sequence. Okay, so the Q1 value of 678.83 and the Q3 value of 531.29 make one transition. Likewise, the next value of 678.83 and 757.38 make the second transition, right? So that is how you see 4 transitions. In addition to the term transition, you also come across the collision energy here, which is 26.4 in this particular experiment, and then the important term of dwell time, which you can see here is 250 milliseconds. So dwell time is the time taken by the instrument to monitor or capture detect the particular fragment, right? So in this example, we are allocating 250 millisecond time to monitor this particular fragment from the experiment, right? When you add up all the dwell times, you come across we come up with a term called cycle time, which in this example is 1 second, right? So the instrument has taken 1 second to note each of these transitions in one round, right? So the instrument doesn't stop by taking just one value. So the dots that you see here on the x and on the plot here are represent one single data point for a single transition, right? Likewise, as the peptides elute over a period of time along the as per the retention time of the peptide, what we see is that there is an increase in the number of data points which get captured. Okay, and the output that you see is something like this. So the curves that you see are nothing but the plots that the data points which have been acquired, they are just joined, and you see a smooth plot as seen in this example, right? So I hope this has clarified the principle of the SRM experiment for you. So this slide gives you an overview of the typical steps involved in a SRM experiment. So you start with sample preparation, and it could be any technique that you choose, be it in solution digestion, in gel digestion, or fast, or whichever technique you choose. But the prerequisite is that it needs a very clean sample which has been spiked in with a heavy peptide if possible. Okay. We then move to the software Skyline. This is an open source Skyline and very widely used for MRM experiments. This is compatible with almost all the vendors. So it is not vendor specific, or it is not biased towards a particular vendor. So this software alone can help you create a skyline, an MRM method, and it can also help you in setting up the experiment, refining it, and then analysis of data once the entire data has been acquired. So the different steps on the mass spec side involve the optimization of the run. So this could be optimizing different parameters like the gradient that you use, the LC gradient that you use, the collision energy that you use, also the dwell time and the pause time, the cycle times that you use. So these are the different parameters which need optimization. Then once the data has been acquired, you analyze it using any software. In this particular case, we would show you how Skyline can be used. So this is just an output of how the data looks like. But then we realize that it would be easier if we show you how the data is analyzed. So I would share a particular example here for a protein called apolipoprotein E, that is ApoE. And this is a differentially regulated protein which has been observed in many diseases like malaria, dengue, then even gliomas. So in the particular example that I'm about to discuss, we have taken our sample of interest. I would not disclose the sample here, but then it has been spiked with synthetic peptide, three synthetic peptides for the same protein. As Meher had mentioned, the synthetic peptide is chemically similar. It is similar in the physical properties as the peptide that is found in my sample of interest. The only difference, however, lies in the fact that there is a heavy label attached in one of the amino acids. In our example, it's arginine. So using Skyline, we now show you how the method has been set up and how the data appears, how you can refine your experiment, right? So let me start with a black document first. This is how the Skyline user interface looks like. It's very user friendly. So this is where I type the list of targets. So I have copy pasted the list of targets for this experiment, and they are basically three peptide sequences, as you can see here. There are different ways in which you can do that. You either take the peptides from your shotgun data and just copy paste them, or uh, or you can use the FASTA sequence from UniProt and just copy paste them as I would just show you in a minute from now. So this two sequences here. This is the third sequence that is being copied. Yeah. So you now see three sequences from for FOE, and let us take a look at the different settings for this. So you now set up the experiment by choosing the settings here. So you can see there are different tabs when you open the peptide settings. This is where you specify the enzyme that was used, the missed cleavages that you expect. It's good that you keep it to 0 in a targeted experiment where you don't want there to be any missed cleavages. Then there are different tabs which can be used depending on the need of the experiment. So I'm just quickly going through each of these tabs. And if you see here, I have selected the isotope label type as heavy. So the heavy label stands for is usually the one which is present in the synthetic peptide that has been spiked to your sample, right? And the modification that I've specified is the one which you see here. So that is how we got our peptides labeled and modified from. And yeah, so I check it, and then there's a quantification tab where there needs to be no change made for this particular experiment. So I say okay, and then I go to these settings again. There's another tab called the transition settings where I would export all the transitions for these peptides based on I would specify some parameters and then get the transitions exported. So here you see the precursor charges. I have set them to be all +2 charge. So in all the precursors that you see, all the sequences that I've selected are the ones which are doubly charged in this case. Then the ion charges, that is the fragments which arise, I have selected them to be singly positive, singly charged or doubly charged. The ion types are Y and P. Here I go for ion 3 to last and minus 2 selection. It depends on which I'm more comfortable with, but it is advised that you don't choose the extreme ends of your peptide. Uh, then the instrument parameters, these are, we use the default parameters here. And then we move to the last, that is the full scan. So there are no changes to be made. This is pretty much the same default method that is used. So I have now made changes for the transition settings. Now, once this is done, I would want to export them, export all the parameters as an Excel sheet, which can be used to give instructions to the instrument, right? So Skyline would help you generate. So you now export the transition list, and you specify which instrument you are using. And we use the thermo quantiva, so we specify thermo quantiva here. And also, if the number of transitions is more, and if your instrument is not capable of monitoring those many transitions in a single run, Skyline gives you an option to select between select the number of method files that you want to create. You could create one method per protein, or you could just create one single method or multiple methods. In this example, we have just given one method. And I say start and end retention times. I would use the start and end retention time for this run because I don't know when my peptide of interest would elute out, right? So I just say export, and I say no, and I just save this as test. So I go to desktop. So you can see that this file contains the information required for the experiment, that is the sequence plus 2 charge sequence. Then you also see the start time to end time, it is 30 minutes. And then the polarity is positive, the precursor charge is this, and then the product arising from these precursors. So these are the same set of precursors. Okay, and you can see that the Q1 value is same whereas the Q3 value is changing, right? And yeah, so this is the basic input file which will be imported onto your instrument, and you acquire your data. Once the data has been acquired, uh, we go to the Skyline file. I'll show you how the analyzed file looks like. So I now import the results from the run. Okay, I now import the results from the run, and you could see I have actually made a sample file ready for this step. So I have imported the result files. Yeah, so I have imported the result files here, and you can see that just a minute, there seems to be a technical error. Just a second. Yeah. So this is how you see the result, right? So there are 3 peptides in our experiment, and if you just click on this plus icon here, it helps you drop down on the sequences. So the Skyline software very clearly mentions that there is a heavy peptide here, right? So this is the spiked in peptide, and the one above, that is 865.9258, is your peptide of interest from the sample, right? And this is how the chromatogram looks. The top view is that for the peptides from your sample of interest. The lower view here is for that from the heavy peptides. So when I click on these, it zooms into the image. So as you can see, there are different transitions which have been detected here, and I can go and monitor the area under the curve for each of these transitions. And when I report the levels of this protein, I take the cumulative value of all the peak area under the curve for all the transitions. Right? And this is how good the heavy peptide looks. Right? The important point to note here is that the heavy peptide gives you a measure of, or gives you an indication as to where your sample is likely to elute. So if we look at this the above sample, you see that it is very noisy. The data is very noisy because there are different other peptides which are also present. The matrix for this is very complex, right? But the heavy peptide, however, would show you a single peak. So it, in some ways, the heavy peptide helps you detect the right peak for studying your protein expression. Right? So this gives us an overview of the software Skyline, and this also brings an end to my topic of discussion today, that is selected reaction monitoring. I hope I have done a job to help you understand this. If not, we are always available over email and the portals where you can post in your queries. And I now request my colleague, Mr. Vipin Panwar, to take over from me and help you understand the concept of parallel reaction monitoring, which is an advancement over SRF, but it uses a different platform about which Vipin would enlighten you. Thank you.
Yeah. Hello. So if I will summarize, like still now you have seen like how we can prepare the sample for the target protein, how we can use the heavy labels to find out the your actual peak, and then you can find the retention time, then you can see the peak area. So you can how basically use. So now I will continue with my another targeted proteomic approach, that is PRM. So PRM basically parallel reaction monitoring. And what are the difference between issue between the PRM and the SRM? So now if you have seen, I just go like difference basically now in SRM. So this as Titan has already explained, that is a selected reaction monitoring, right? Now if you have some peptide, like there are some differentially active peptide which you want to monitor and then you want to quantify how those peptide is like is in your samples. So you put your method, and then those peptide or precursor ion will pass through the MS1 filter, and then they will fragment here in the collision cell, and then again they will be scanned into MS2. So in MS2, basically the specific transition will be monitored only, like which we already put in our method. So only specific transition will pass through this particle 2, and then they will be detected. So here you will get a, like different transition. Now, in case of PRM, that is a parallel reaction monitoring, what is happening here now, we are selecting our precursor and then we are injecting into the mass spec. So they will pass through this particle and they will, like all precursor ion will be fragmented into a collision cell, and they will move into the orbit ref or the time of flight. So the main difference here is now those fragmented ion we are not going to monitor. So we are not saying like this fragment have to be monitored. So all fragmentation ion have to be will be monitored in top and orbit ref. Now here you can see the all transition, all fragmentation ion you can see. Now in SRM, basically you are giving a very less window like zero point 0.7, but in case of PRM, you can put like window isolation window, which is in MS1. In both case that is quadruple. So in case of PRM, you can put around 2 M by Z also. So and if you say in this case SRM, so the hardware selected MS2 fragment, because now here we are selecting which fragments have to be monitored. So that is selected by quadruple 2. But in case of PRM now, the software selected MS2 fragment. Now all fragments I am deselected, that's why it is full scan MS2. Now how it is done, like how it is working PRM, if you see now these are the peptides which is going to the mass analyzer, basically which is quadruple for PRM. Then those peptides will be fragmented into a collision cell, and those fragmented ion will go into a mass analyzer. So here in mass analyzer, you can see all fragmented ion will be detected and they will give a spectrum. How the data points will be collected. Now, there are different instrument having a different scan rate. Like if I will talk about QE, so it having a 7 hertz. If I will show, so if I will say about QE, so if I say like QE, so see if we like put the resolution around 35,000, so it having a scan rate 7 hertz. So 7 hertz means 7 peptides can be monitoring within 1 second. So accordingly, we have to adjust the timing also, and then cycle time also. Now, if you go again on slide, now if we are giving around 7 peptides, now we are giving around like 7 peptides now. So after each seconds, again those peptide will be monitored and then they will give a data point. And then this data point will make a peak. So on the basis of you can select your peptide, so you can see the peak response for your fragmented ion. And in case of PRM, because all of the fragmented coming together, so they will be a co-eluted or fragmented ion. Okay. So generally for any proper quantification, we keep around 8 to 10 data points. Actually, if you see in internal part of the QE, if you see how the peptides and the precursor ion will be fragmented, and then how they are analyzing in orbit trap or into so. So first, there is the first particle where the isolation window where we are filtering our precursor ion. So once we said those precursor ion have to be passed only, so those precursor ion will go and in collision cell and they will be fragmented there by using different collision energy. So generally we keep 25, 27 collision energy. So once all peptides will be fragmented, then those peptides will go into a orbit trap, not this orbit trap, and they will be analyzed here and then detected. Now here you can see like both analyzer and the detection within orbit trap is there. Now once you know, now you get the peptide peak and then the peak response. Now how you verify this is your exactly your peptide peak. The first, if you see, now all transition come together, which show the coalition of concurrent transition. So for a specific peptide, all transition the fragmented ion will be come together. Another you can verify your sequence, so you will get the Y ions, the Y ions and the ions and by G of those. By using that you can do the sequence query also. Third is you are using a reference internal standard which we already saw during the sample preparation. If you are adding some heavy peptide, so if heavy peptide is also coming on the same retention time as you are interested in peptide or your target peptide means you are getting a right peptide. Now if you see here how you they quantify, like here they are taking only 4 data points. So in case of it is like if your peptide is very less, so it will give a very less response as compared to disease. 4 data points. Now in the score, as Saitan has already explained about uh, skyline, how we generate. So the main difference in PRM method and SRM method is in case of SRM we are exporting transition list. But if you have to do PRM, then we have to export isolation list. If I will open a same file, the setting of the peptides and those will be approximately same. We don't need to change. So it's already explained by Saisran. If you see those settings, digestion, prediction, filter, library, modification. So most of the settings will be a same in case of PRM also. Thus, when you are going to export those, whatever we are giving or have been analyzed in orbit ref and top. Now, once we have created this isolation list, like it will be like that, then we get this list and then we upload in the instrument. And then instrument during the instrument uploading, then you have to set those parameters. This is like full MS parameter. Because here it is first we have to do QC check of your sample. So we are keeping very low resolution. You can see 17,500. And then after that you have to do PRM of them. Then here you are increasing the voltage, sorry, resolution. So here is around 35,000, and then you are giving a time to acquire all fragmented ions around 120 milliseconds. So these are just MS parameter and the parameter. So once we run the samples, then we get this type of file, and then we get those raw files. So once we get raw files, then we have to again analyze into skyline. So I will go again skyline. So we open the same file and then we import our data, which has been run. I can import the same. This one. I'll just now define where it has been saved. Desktop. So now all files which we have run already on QE, so those files will be imported into Skyline, and then we can see the how let it import. Now once let us assume we already run those 3 files and then we are importing into Skyline. It will take around 1 minute. So once we import the skyline, then we have to see how the peak shape is visible, it is properly coming or not coming, and how the retention time is matching with your heavy peptide. So that you have to see. Now it's already important. Then I will just see now. Now I will go to setting and then view range graph file. Let's read extra one. One. Now, yeah, here now you can see the above one is the peptide which is peak is coming from your samples. These are like fraction sample 1, sample 2, and these are sample 7. So this above one is the peptide peak is from your sample, and lower one which is from your heavy peptide. Now here you can see the transition. If you see the retention time of both of them is same. So here we can confirm whatever the peak we are getting at the exact at retention time. Now if you want to see the now if I show the retention time also, you can see how it is looking in sample wise. Like this is a peptide, this is for this I think same, right? This is for this is for another sample. So if you see the sample 1 and 2 or 3, it is retention time is same in all heavy and then your interested peptide. In a similar way, if you want to see peak area, how it is different in different replicates, so you can go peak area and then replicate complete. Now if you see now here 1 or 2, so this is 1 and 2, so this is from your samples and this is from your heavy peptide. So in this way you can confirm like whatever you peak you are getting, it is exactly from your peptides or no. So I'll go to the slide. Yeah. So hope now I hope you know now you get an idea about the mass spec and then what are the different techniques we can use to do our experiment, and then how you can now if you have some you got some like different some targets and how you can validate by some different approach. Okay. So hope now you done. Now next maybe in next deep live session we will do on microarray. Thank you.